Hello dear students, welcome to Practical Biochemistry. The topic is estimation of serum total protein by Biuret method. Estimation of total serum protein. Why this is required? Because it is useful for checking any major change in protein level due to any disease. Except immunoglobulins, majorities of plasma proteins are synthesized by liver. Various tissues catabolize plasma proteins. Estimation of total serum protein is an important test because it reflects the concentration of plasma protein between their synthesis and catabolism. What is the principle of this method? So Biuret method is also known as Piotrowski's test because it's documented by Gustav Piotrowski, a Polish physiologist, in 1857. Now, as per the principle, proteins, when it reacts with the cupric ions in the Biuret reagent under the alkaline medium, it gives purple-violet color complex. And this purple-violet color complex, absorbance of which is measured at 540 nanometer. This reaction occurs in compounds that have at least two, this type and this type bond or similar group attached directly or via a nitrogen or carbon atom. So that is the principle of this Biuret method. Which are the reagents required for this test? Biuret reagent, total protein standard, which is 6 gram per 100 ml solution, that is 6 gram per dl. It requires test sample, that is the serum from plain bulb or the vacuity. Now the procedure. Take 3 test tubes. Label the test tubes as blank, standard and test. Now add 1 ml of Biuret reagent in all 3 test tubes, blank, standard and test. In the standard test tube, add 10 microliter of the standard solution. In the test tube, add 10 microliter of the sample. And in blank test tube, add 10 microliter of the distilled water. Now mix well all 3 test tubes. Incubate at room temperature for 5 minutes and read absorbance colorimetrically at wavelength of 540 nanometer. Now the procedure part. Take 3 test tubes. Label the test tube B for blank, S for standard, T for test. Now add 1 ml of Biuret reagent in all 3 test tubes. Add 10 microliter of distilled water in blank test tube. Add 10 microliter of standard solution in standard test tube. Add 10 microliter of the serum sample in test tube. Mix well all 3 test tubes and incubate at room temperature for 5 minutes. After 5 minutes, take reading colorimetrically at wavelength of 540 nanometer. Observation. OD of blank 0.05, OD of standard 0.13, OD of test 0.10. Now the calculation. OD of test minus OD of blank divided by OD of standard minus OD of blank multiplied by concentration of the standard. So for this calculation, the values 0.10 minus 0.05 divided by 0.13 minus 0.05 multiplied by 6. So ultimately it is 0.05 divided by 0.08 multiplied by 6 is equal to 3.7 gram per dl. Concentration of total protein in given sample is 3.7 gram per dl. Reference range for serum total protein is 6 to 8 gram per dl. So here there is decrease in the total serum protein level. So this condition it is known as hypoproteinemia. Now the interpretation. If the values are increased, then the condition is known as hyperproteinemia. Which are the causes of hypoproteinemia? Hemoconcentration in dehydration due to inadequate water intake or excessive water loss as seen in severe vomiting, diarrhea or Addison's disease. Disease of reticuloendothelial system. In multiple myeloma, tuberculosis and Kala Azar due to increased synthesis of gamma globulin fraction. In collagen disease, in autoimmune disease, for example sarcoidosis and rheumatoid arthritis, we found that increase in the total serum protein level, the condition is known as hyperproteinemia. Hypoproteinemia, the causes include hemodilution due to water intoxication or salt retention, impaired protein synthesis, for example in conditions of liver disease, malnutrition, malabsorption, hypothyroidism and wasting disease, congenital disease like agammaglobulinemias, loss of protein from the blood, renal disease, this includes nephrotic syndrome, chronic glomerulonephritis, metabolic disorders, these include diabetes mellitus, burns involving larger portion of the body area that is due to loss of plasma from the damaged capillaries. So all these are the causes of hypoproteinemia. Now what is acute phase proteins? What is acute phase proteins? Proteins that change their serum concentration by more than 25% in response to inflammatory cytokines like interleukin 1, interleukin 6, tissue necrosing factor alpha. The proteins that change their serum concentration by more than 25% in response to inflammatory cytokines, these proteins are known as acute phase proteins. Acute phase response is mediated by innate immune system and responsible for fever, leukocytosis, increased cortisol, decreased thyroxine level, decreased serum ions, etc. Now there are two different types of acute phase proteins, positive acute phase proteins and negative acute phase proteins. Now which are the examples of positive acute phase proteins? These include C-reactive proteins, serum amyloid A, heptoglobulin, ceruloplasmin, alpha-2 macroglobulin, alpha-1 acid glycoprotein, fibrinogen, complement C3 and C4. So all these are positive acute phase proteins. Now which are negative acute phase proteins? This includes albumin, transferrin, transthyretin, retinol binding protein, adiponectin. So all these are examples of acute phase proteins. Now which are transport proteins? Proteins that helps in the transport of various substances and ions across the cell membrane, and this called transmembrane proteins. It includes ion channel proteins, carrier proteins, sodium potassium pumps, GLUT1, glucose transporter 1, proton pump, etc. So all these are the examples of transport proteins. If you like this presentation, please try to share it with your friends, group, batch and colleague. Thank you so much everyone.